Now, coming to the next part of the immune system, which is very important, is the organs of the immune system, the lymphoid organs. We all know that there are two categories of organs. If we divide the immune system very broadly, the primary lymphoid tissue consists of the bone marrow and the thymus, and the secondary lymphoid tissue, which consists of the lymph nodes, spleen, the malt, and the salt. Now, this is a picture which actually depicts that how the lymphoid tissue are organized within our body. They can be present in the form of a diffuse collection of few lymphoid cells as we get patches of these cells in the intestine. They can be present in form of follicles. They can be present in form of patch, if you remember the pious patch. So they can be present in form of patches, or they can be present in the body in form of a well-defined organ. So this is in fact the order of the increasing complexity in which the lymphoid organs are present in our body. Now if we move on to the next particular diagram, now please concentrate on this picture. The black, I mean to say the black color, the black font actually indicates that these are the antigen accessing or inductive sites. Now, what is an inductive site? So we have inductive sites here. So these are the various categories of this is the Galt, and this is Nolt or Walt, and this is the urinogenital system. So we have some inductive sites in each figure. An inductive site is a specific area in a mucosa where an antigen is encountered, where an antigen is encountered and primary adaptive response is initiated. For example, inductive sites in the Galt includes the organized lymphoid structures in the small intestine called Peyer's patch. I just now said that. The appendix, very few of us know that the appendix is also a part of that inductive site, the appendix. And diffuse collection of lymphocytes and antigen presenting cells scattered within and just under the gut epithelia. In the Balt and the Nolt, the main inductive sites are the bronchial epithelium and collection of tonsils in the nasopharynx respectively. So, from the definition of the inductive sites, you can assume that these are the sites where the antigen accesses the body or where the antigen enters the body. Now, if these are the sites where the antigen accesses the body, then there are sites which will produce effector response to the antigens that have entered. Now, look at the blue colors. The blue colors are the areas. So, we have this blue colors areas which provokes the immune responses. For example, so the blue colors are the effector sites. A mucosal effector site is a specific area in a mucosa to which effector lymphocytes are dispatched. Among the most important mucosal effector sites are the exocrine glands such as saliva and the lacrimal glands. These glands produce protective external secretions that contain antimicrobial molecules and secretory antibodies. So, this picture of the mucosal system, we saw that we are having two sides, the black one where the antigen is accessed, pars, patch, appendix, small intestinal mucosa, these so on. And the blue areas are areas where once the antigen are accessed, the antigen is recognized and effector response is built up, and then the effector response is built up, and then the molecules put up a challenge against the antigen. So, since I have delved in detail with the bone marrow and the thymus, we had studied in detail the development of T lymphocytes through the thymus. So I have not gone into the summary or the revision of the bone marrow structure or the thymus structure, even though I'll show you a structure of bone marrow later on, and I'll go over a brief summary of the B and the T cell development, but right now I'm concentrating mainly on the secondary lymphoid structures. Now, this is a structure of a salt. I wanted to show this picture because at the initial stage, whenever an antigen enters the body, what happens? What happens? Actually, what happens? What is the step that follows? So, coming to this, if you look, a pathogen is entering through the epidermis. Now, this is the skin. This is the epidermal area, and this is the dermal area. The thin line is the basement membrane. Whenever a pathogen is entering the epidermal area, the keratinocytes present here, they are activated by the pathogen, and the immediate keratinocytes present here gets activated by the pathogen, and they start liberating two quality pro-inflammatory cytokine, the IL-1 alpha and the TNF, which further activates the adjacent keratinocytes. The moment the adjacent keratinocytes are activated, as you can see, a host of other chemokines and cytokines is secreted. So this is happening on one hand. On the other hand, whenever the pathogen is invaded, the antigens of the pathogen, if fragmented, the antigens come out, and they are then recognized by, you can see this category of cells, the gamma delta T cells. So these category of T cells, I was just speaking in my earlier recording that these category of T cells are very important in the mucosal immunity. They are found in great numbers in the skin, and they play a big role out here in the mucosal immunity. So as you can see, the gamma delta T cells, they do not need antigenic peptide complex with MHC. As I had said in my earlier recording, they can directly bind with the antigen. You can see here the macrophages are activated after capturing the antigen, and you can see the Langerhans cells are activated after capturing the antigen. Once the antigens are captured, as you can see, now what we are seeing that the matured and the Langerhans cells then activate the matured dendritic cells, or the Langerhans cells at times get further modulated by the antigen capture to become mature dendritic cells, and now they present antigens, as you can see, to the alpha beta T cells if there are any memory population present in that position. Okay, so we are getting the effector alpha beta T cells. So we are seeing that in case of immediate infection through the skin, you can see that the initial protection is taken up by the gamma delta T cells, and later on it is taken up by the effector alpha beta T cells. Now let's look into another picture. Now if there are antigens, the antigens are further captured by the Langerhans cells. The Langerhans cells can modulate on antigen capture to get converted into mature dendritic cells, or the Langerhans cells can move in through the afferent lymphatics into the lymph nodes, where they can present antigens to the naive alpha beta T cells. You can see, and this naive alpha beta T cells are activated. Interesting thing is that once these naive alpha beta T cells are activated and they produce various kind of progeny lymphocytes, you see there is a CLA marker in each of the lymphocytes. Now what is this CLA? This CLA is nothing but these effectors are induced to express cutaneous lymphocyte antigen, a sialylated carbohydrate that specifically binds to E-selectin expressed on the post-capillary venules of inflamed dermis. So the moment the CLA expression is occurring, through the E-selectin, they move through the blood vessels, and via the E-selectin, these activated cells can migrate out from the venules. So the main role of CLA expression is thus to allow the T cells to migrate through circulation and go to sites of inflammation in the dermal region. So this is the first thing that happens whenever an pathogen enters through our skin. So these are the variety of cascades that takes place whenever a pathogen enters through the skin. Now, next, moving on to another important. Now, I'll not be getting into details of these two particular because I talked in detail about one lymphoid organ, secondary lymphoid tissue. This is the structure of gut, as you can see. But one interesting thing I want to show you, if you look into the gut epithelia, there is a muscle underneath muscle layer. We have the lamina propria and we have the epithelial layer. And you can see the epithelial layer is folded into crypts. We have crypts here and we have the villi. And another important thing you should note, that you can see the presence of the gamma delta T cells interposed between the villi cells. So these are known as intraepithelial lymphocytes. Another important thing to note is that on the surface of the villi, we are seeing two categories of I'm going to say immunoglobulin IgA, the same category of IgA. So one is the SIgA. See, S is small and the other is the SIgA, where the S is capital. Now, the SIgA, the small one, this is the secretory IgA that has been already secreted. And the other one, the capital one, SIgA, this is the molecule which is to be secreted. So there's a difference. One is a secretory IgA and the other is the secreted IgA. So this has been secreted out and this is on the verge of secretion. Now, what's the difference? The secreted Ig lacks the transmembrane and the cytoplasmic domain, but does have a tail piece. While the secretory immunoglobulin is formed when monomers of secreted immunoglobulin first polymerize around the J chain protein and then acquire a secretory component while passing through a mucosal epithelial cell or a glandular duct cell. So we have the presence of that J chain secretory component. So this is one difference I wanted to show. And another thing, if you see that I talked at the initial stage in my class of the natural killer T lymphocytes. So you can see the presence of the natural killer T lymphocytes. Then we have the variety of cells in the villi, the goblet cell, the Paneth cell, and another important thing, we have certain cryptopatches in the lamina propria region. So these are known as cryptopatches. They are basically, as you can see, the composition of the cryptopatches. The cryptopatches are mainly made up of the alpha beta intraepithelial cells and the gamma delta interepithelial cells. Okay. So these are they are present as cryptopatches, and if the need arises, this cells gets activated, comes out from the patch and then can interact with the antigen. So this is a very basic structure of the gout. And now coming to one important essential structure, the vault or the knot. Now, given the COVID scenario, this passageway has become very important, and you can see that we have, if you look into the structure, these blue structures are the pharyngeal tonsils. So we have a lingual, a palatine, and the pharyngeal tonsils. So these are the structures of the tonsils. And this is the structure of the airway. We have the submucosal gland, the trachea. Here again also you can see the gamma delta T cells. So you can understand the importance of the gamma delta T cells. They are present on the mucosal surfaces, providing a basic first-hand adaptive immunity to the mucosal surfaces prior to the incoming of the alpha beta T cells. Then we have the M cells. Now, one thing, interesting thing of the M cells is that the M cells helps in transcytosis of antigens. So if there is any antigenic protein passing through the airway, the M cells play a role of transcytosis through of the antigen, and once the antigen is passed on, once the antigen is passes through the M cells, enters, then the antigen capture is done by immature dendritic cells. The immature dendritic cells get matured and they might produce a Thi 1 response, a T helper 1 response, or a T helper 2 response. Now, if the antigen that has been transcytosed by a M cell is weak antigen, usually a T helper 2 response is developed. Remember, It's a weak antigen. It's a T helper 2 response, which further activates the B lymphocytes. Thereafter, the B lymphocytes, on recognizing the antigen, actually produces the epitope against the, actually produces the antibody against the antigenic epitope. But if the antigen is strong antigen, then the dendritic cells are activated and produce IL-12 if it's a strong antigen. Once IL-12 is produced, T helper 1 is activated, and the T helper 1 then activates the cytotoxic T cells, which then results in the granzyme-mediated lysis of the infected cells, if any. So it is the M cell, depending on the antigen signal received by the M cells, it depends that whether the B lymphocytes will be activated, because for the activation of the B lymphocytes, if it's a weak signal, the cytokines involved are IL-4, IL-5 and IL-10. If it is a strong signal, the cytokines involved are IL-12, and that actually activates the cytotoxic killer cells. So this is roughly about the NALT or the BALT.